So as you know, I do work with maize. I've worked with sunflower. I love being out in the field. But today, I'm going to take you guys for a little field trip. We're going to get out of the field. We're going to go head over to the forest. So today, I'm going to introduce you to a suite of tools that I have called CONIFER. It is, maybe, not going to the next slide. There we go. All right, so CONIFER is a suite of tools that was developed for the analysis of archived genomic data and the extraction of individual samples from megadata sets. It stands for Converting Old to New, Individualized FASTA and FASTQ Extractor for Researchers. And it features, oops, sorry, this is not responding very well. It features automated data processing and email notifications. So it gives you a very easy, automated way of running your data through your pipeline. Now, our objectives that we established when we went around thinking about this pipeline were to, one, improve accessibility of archived sequence data, to enable meaningful conclusions to be drawn when comparing data across multiple studies, also to be able to extract sequence data for individual samples, combine sequence data from multiple studies to improve sequencing coverage, and finally, but very importantly, to provide a user-friendly sequence data analysis pipeline. Because that's one of the big cumbersome things for data analysis is figuring out, how do I even start? I know nothing about any of this. And many grad students, that's where you start. Even if you're not a grad student and you move into a new field, that's where you're at from the get-go. So I wanted something that was going to facilitate that. So CONIFER was born. Now CONIFER is made up of two modules that operate within a pseudo GUI, so that's graphical user interface. And the first module takes and converts your SRA files to FASTQ format. So that's taking it from an archived format into something that you can actually use for your research. It also prepares sequences for use in Tassel. For those of you familiar with Tassel, it is a pipeline put together primarily for GBS data, but flexible enough to handle other genotype data as well, to give you those genotype calls. So module 2 also comes in. This gives you that option for uniform quality control and genotype calling parameters to meet those three middle objectives there. And it also gives you the option for data subsetting and multi-source concatenation. What I mean by that is taking that data from several different studies, pulling it all together and getting that overall better coverage of your genome. And finally, we have the Tassel pseudo GUI. And what that is, it's kind of a hybrid between the command line and your traditional point and click GUI. So you're not going to be in there with a whole bunch of JavaScript with those awesome gray boxes where you click OK or cancel, but it is going to prompt you and say, what file would you like to use? What brands would you like to use? And it saves them for you. So it automates the process. So some of you may be wondering, well, what is the sequence read archive and why should I care? Especially right before lunch. I mean, come on. So the sequence read archive was established back in 2009 in response to the just mass of genomic data that we've been generating since the next generation sequencing revolution. And as you can see on the graph in front of you here, its growth has been almost exponential. And it shows no signs of stopping. We continue to generate more data. We're not going to stop generating data. We're not going to sit on our hands. We're going to keep doing research. In the meantime, we have this awesome treasure trove of data just sitting there, just waiting for us to delve into it. There we go. All right, so to give you a better feel for what the SRA is and how CONIFER can help you make the most of it, I'm going to give you a drone's eye view of the situation. I'd like you to consider the SRA as a forest made up of rows of trees, and within those rows, individual trees. So if you pull it back and you look at just a single growth, consider that as a study. You upload individual studies up to the SRA so that it can be stored for use at a later point. Within those rows are individual trees. Those are your individual samples that fall within a study. Now, currently, the way that it's set up, you can download an entire study but it's very difficult to subset that data out. If you want a list of sequences for a single sample, it's very difficult to do, and there's no infrastructure set up in order to handle that. So CONIFER lets you do that. And I apologize, it looks like that shifted over a little bit, but so I gave you this decision tree here. I'm not going to sit on it for very long because I'm going to bring it back in a little bit. But just ponder while we're going through this, why would you want to use CONIFER? Is it something that you can apply in your lab? Is it something a coworker can apply in their lab? So first, we got to tackle the SRA. It can be complicated to navigate, but I just wanted to give you a quick run through. You identify a study of interest. The publication will contain the study number. You plug that in. I highly recommend using FileZilla. You go and you pull out the study, you put it onto a working directory, wherever it is that you're going to want to do your data analysis. Then you can run it into CONIFER, sorry. So at this point, the question of whether or not you're using archive data is yes. You wanted to pull something off the SRA, it was interesting, you didn't want to have to resample it yourself. So we are going to go through Module 1. Now what Module 1 does is it takes and transfers your files, after you transfer your files off the archive, it takes and reconfigures the folder structure in order to give you an efficient means of converting that data from an archive format into something that you can actually use. There is a tool within the SRA toolkit that you can download, and it does it. The folder structure as it stands is very inefficient. So all this does is it lumps all of those other subfolders up into one header folder and allows you to run everything in one fell swoop. So at this point, if you have a favorite genotype caller, your data is ready to go. You have your FASTQ files, you're set. But if you want to use Tassel, or you're looking for something that's a little bit more user-friendly than what is currently available for you, hold on, we're going to go on to Module 2. Now, Module 2 is what I talked about before. It allows you to subset your data. And what I mean by this is you can pull out those individual samples within a larger study. So say you have 100 samples, you're only interested in 5. This will pull out those 5 and give you 5 separate files with that sequence information. So essentially, you run it through the first part of Tassel using the pseudographical user interface. Very automated process. It generates this matrix that gives you total counts of the sequences. You've undergone a bunch of quality control steps at this point. This is all built into the pipeline. And all this does is it merges your replicate samples and converts them down into a binary format so that the computer can go, okay, presence, absence. Do we have the sequence? Do we not? If we have the sequence, we're going to output it to a separate file. It's going to give you that single individual file for a particular sample containing all the sequence information. And for some applications, FASTA isn't good enough. So you can also go back in and pad your FASTA files with quality scores to generate FASTQ. At this point, you've already done your quality control. So the actual quality scores aren't important as long as they still meet that criteria that you would have set otherwise to pipe them through, you're set to go. And at this point, again, if you have a favorite genotype caller, you can use it. This pipeline is flexible enough that you can pull it out at whatever point you want. However, if you decide you like the pseudographical user interface, you can continue on and run it through Tassel through to the end. So again, is CONIFER for you? Are you using archive sequence data? Yes. Well, then you're going to want to execute Module 1. It'll get that into a format that you can use in a very readily. If you want to subset it, you can pass it through Module 2. If you don't, that is perfectly fine. You can continue on using that pipeline or your another sample caller that you have a preference for. However, the beautiful thing about this is that old is a relative term. It can be something that somebody else published and uploaded 5 years ago, or it could be your data that you just got back 5 hours ago. You can use old data, archived data, brand new, hot off the press data. It's flexible enough to accommodate for all of those. So with that, I would like to acknowledge my lab, my committee members, and the Buckler Lab for putting together the Tassel tool and for answering many of my questions. And also technical expertise, in particular, Nathan Weeks, who, without whose help, I probably wouldn't be up here standing telling you guys about this pipeline, because when I came in, I knew it was a black box. I knew that much about computer scripting. And so to come from knowing nothing to being able to offer a tool for other people who are starting in that same place that gives them that step up into data analysis, I'm very happy to take questions about the pipeline, whether right now or over lunch hour, because I know you guys are all getting hungry. So with that, I will open up the floor to questions.
Thank you for that really good presentation. My first question is basically, how do we access this pipeline? Where is it at? Is it something that is web-based, or do we have to download it on a computer?
Yes, so this is something that is currently in production. It is not quite at that point of release yet. It will be within the next year, hopefully sooner. I will be uploading it to GitHub, and if you want to be put on a list to be notified about its release, shoot me an email or find me on LinkedIn, or you can contact me through my personal website as well. But what it is it runs in a Unix environment. So that makes it something very awesome to use if you have computer clusters. You want that extra space, you don't want to run something on your desktop, you don't want it to take up valuable space there. You can run it on a remote server.
All right, please say thanks to Hannah.